Hello everyone. In this video we're going to be talking about something called the mark, release, recapture method. Now this is a technique that's employed when trying to sample to work out the population of a mobile organism. Now typically in GCSE and both A-level biology you look at ecology and methods of sampling, and you talk about how you can sample things that don't move via the use of a quadrat, like plants for example. And then you look at mobile populations and you think about maybe the Tulgren funnel, pitfall trap, pooter, those kind of things. But this is another sort of method that is taught. Now, the first thing is that we need to appreciate that different sampling methods are more appropriate than others depending on the species or habitat that is under investigation. So this would be used for certain organisms if it's a particularly large population. So mark, release, recapture, often actually just shown by its abbreviations, MRR, for mark, release, recapture, not to be confused with MMR, the measles, mumps, rubella vaccine, which I have seen students do in the past. So the mark, release, recapture method, this is how it ultimately works. Using this technique, a large enough sample of animals from a population is caught, is counted and marked in an appropriate way. So let's just say, for example, so we're going to just set a bit of a scene. Let's say here we've got some grass. Just a very quick sketch. Let's say we're outside and we happen to see there's a good number of snails. I mean, this works with any kind of sort of large population. Now, it's not the best drawing of a snail, but it'll serve this purpose. So let's imagine that we go out and we're trying to calculate the population of snails in a particular habitat. So we're going to go out, let's draw some sky here, just to make it a bit more of a picture. So we're going to go out, we're going to mark the snails that we catch. We're going to release them back into the habitat and wait a given time period. Then after a suitable length of time, we're going to go and catch a second sample. So we catch one group, if you like, we mark them, release them, and then after a suitable length of time, we go catch a second sample and we count the number of animals that are marked that we caught this time. And this whole procedure uses something called the Lincoln index. So that's what we're going to work to. Now the Lincoln index is a formula that allows you to sort of work out an estimated population size. And this is the formula. It says that if you take the total number of individuals in the first sample, so just to save space, I'm just going to write and abbreviate total number of individuals, we'll just say that's TNI, total number of individuals in the first sample, so when you first go out. If you multiply that, so multiply that by the total number of individuals in the second sample, and then divide that number by the number of marked individuals recaptured. So the number of individuals that are recaptured that are essentially marked. The number of individuals marked from the second sample. And that is what our Lincoln index is. So the total number in the first sample times by the total number obtained in the second sample divided by the total number of individuals marked from that second sample. So let's just use some numbers just to put this into a bit of a context. So let's say, for example, we go out and we catch 100 snails and we mark all 100. And let's say we mark all 100 and then we come back out a few hours later, even a day later, and this time we only catch 80, and of that 80 we notice about 20 of those are marked. Now that would actually, when you do that sum, that would come out as 400. So what that says is that we estimate the population of snail to be 400. Now, very rarely and occasionally, that may actually work out, that may be close to the population. But more likely than that, it may very well just be way off. And it's because we make a good number of assumptions in this whole process. So a lot of the times when you work through an example, particularly an exam question example, the number that you calculate tends to be way off from what the actual population is. And it's because of these assumptions. So what we're going to do is we're just going to talk through actually what we have assumed to be happening in this entire procedure. Okay, so what we're actually going to do is, I think I'm going to make a bit of a tally just to record so you can see how many different things we're actually going to be assuming in this. So I'm going to talk you through all the assumptions. We'll make a little bit of a tally every time that we consider one. So first of all, we are assuming that those marked individuals released from the first sample distribute themselves evenly within the whole population. There's one straight there. Not only that, not only are we assuming that they're evenly distributed, that we give them enough time to do so. So we're assuming that they have sufficient time to do that. That's two. We're assuming that when we go back and collect our second sample, that an identical sampling method is used. Now, that often is kind of a given. We would normally tend to employ the same method, but again, this whole process just assumes that that is the case. We also assume that the proportion of marked to unmarked individuals in the second sample is the same as the proportion in the population as a whole. So we're almost saying that if we include, well, that basically means the marked ones and the unmarked in that second sample, that would represent almost our population. So there's a fourth assumption already. We imagine that the population has a definite boundary. So there is no immigration and no emigration. So nothing is coming in to our environment, our area that we're sampling, and nothing is leaving. So we're imagining that everything is just stationary almost. Now here are two that we most definitely assume. When we do this calculation, we're presuming that there are none, or at very best maybe a few births. Now, you know, when sampling any population of organism, there are likely to be births, but we're going to say that there are none. Equally, we're presuming there's no deaths. So that's 8 presumptions that we have already made about this habitat that in real life just would not be the case. Now what's really important to notice, or to consider rather, is this mark that we're putting on this snail. So this snail at the bottom, we've come along in the first sample and we've put some, I'll just colour it in red, we've put some thing onto it essentially, some adhesive, some mark. Now typically when you do this on a field study, you'd kind of use paint, but we're presuming that, well, first of all, that this mark isn't rubbed off at all during the investigation. So that takes our assumptions up to 9. We're also assuming that the marking method that we use is not toxic. So that would be our 10th. And similarly, we're presuming that the mark does not make that individual more conspicuous and hence more liable to predation. So there is our 11th assumption. So you can see this is a particularly flawed technique. This statistical technique of working out population size, it only gives you a very rough estimate, but perhaps done on a large scale over a repeated sort of study you may end up getting a value that's more reflective of the real population. But this is just one of many methods that we can use in science when sampling mobile populations. Okay, I hope that helps.